Good afternoon to one and all. I, Harshida Gadvi, welcome you all here for the national webinar on Bioinformatics for Healthcare, organized by Gujarat University Botanical Society. We have with us our Honorable Vice Chancellor Professor Himanshu Pandya as President of the function, our Pro Vice Chancellor Dr. Jagdish Bhavsar, our Registrar Dr. P. M. Patel as our guests of honor. We have with us our distinguished resource person, Professor Rakesh Rawal, Professor and Head, Department of Life Science, Gujarat University. Faculty members of the department Arshna ma'am, Solanki sir, Maitre sir, Modi sir, Soumya sir, and participants. I welcome you all heartily. Gujarat University is on a fast track of progress. We are proud of our university, and as a mark of respect, let us rise for our university song.
वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन गुजरात यूनिवर्सिटी आपणी गुजरात यूनिवर्सिटी आपणी विद्यातणु तपोवन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन
राष्ट्र चेतना कला साधना खेल भावना विकसे
विश्व मानवी बनी गुजराती विश्व सकल માં વિલસે સ્નેહ સંપ સહકારની સાથે દેતી તું સન્મતિ સૌના સાથને સંગાથે સૌ સિદ્ધ કરે ઉન્નતિ પ્રેમ બંધુતા સમાનતાના પલ પલ પ્રસરે સ્પંદન વંદન અભિનંદન વંદન અભિનંદન वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन वंदन अभिनंदन
Harshada, you are mute.
Now I request Arshna ma'am to formally welcome the guest.
Thank you, Harshada. Good afternoon. It's indeed a pleasure to welcome you all here for the national webinar on Bioinformatics for Healthcare, organized by Gujarat University Botanical Society. Gujarat University, with the vision and mission of our Honorable Vice Chancellor Professor Himanshu Pandya, is progressing by leaps and bounds. We welcome Pandya sir, who has always inspired not only the students, but the teaching and the administrative fraternity to excel with utmost dedication. We are proud that our university has 5-star ranking by GSIRF and has been selected in top 50 universities in India by NIRF 2021. We also welcome our Pro Vice Chancellor Dr. Jagdish Bhavsar, who has always motivated us and guided us in all our endeavors. We also welcome our Registrar Dr. P. M. Patel. We heartily welcome our distinguished resource person, Professor Rakesh Rawal, Professor and Head Department of Life Science, Gujarat University, and are grateful to him for accepting to share his expertise with us. I also welcome my colleagues in the department, Professor Hitesh Solanki, Professor Bharat Maitre, Dr. Nainish Modi, Dr. Soumya Patel, and all participants. Healthcare has always been a thrust area of research, more so after the COVID pandemic. Integrating bioinformatics research with basic research can not only give us a unique insight into the disease, but also lead us towards its cure. I once again welcome Rawal sir and request him to share his research experiences with us. We look forward to a great learning experience. Thank you.
Thank you, ma'am. Our Vice Chancellor, sir, Professor Himanshu Pandya, is in a meeting and has conveyed his best wishes for this webinar. Our Pro Vice Chancellor, Dr. Jagdish Bhavsar, and our Registrar, Dr. P. M. Patel, is also in a meeting and has conveyed his best wishes for this webinar. We have with us our distinguished resource person, Dr. Rakesh Rawal. Sir has been the backbone of research in bioinformatics at Gujarat University. His expertise in cancer and stem cell biology has led him to publish many research articles. Sir has completed several research projects and guided students in major disciplines in biology. Sir has trained several scholars and faculty members in finishing schools funded by GSBTM. He is a member of Indian Association for Cancer Research and is associated with Institutional Biosafety Committee as DBT nominee. He has been awarded as a Best Professor of the Year at Gujarat University in 2019. Sir, we are very grateful that you accepted our invitation and we look forward to your talk on bioinformatics for healthcare. Over to you, Raval sir.
Thank you, Arshida, for a wonderful introduction, which I never had. Anyway, so today I am here because I had a background of around 25 years in cancer research way back in 1991 to 2017 before I joined Gujarat University. And I have been associated with Department of Bioinformatics since then. And that's how my passion for bioinformatics, even though I'm not a certified bioinformatician by degree, but we were initial few species who were struggling to keep the life of bioinformatics in the curriculum. And that's how my interest percolated, and I tried to communicate and interact with students, and I always learn from them. And I will go through an overlay overlay of the role of bioinformatics in healthcare specifically. So can I share my slide?
Yes, please. Sure.
Hope it is visible.
Yes, sir.
Yes,
It
sir.
is.
Yeah. So, as per the title and as per the subject, the term bioinformatics in a textual format can be described as the use of computational power, where we talk about the IT, information technology, to acquire, manage, analyze, and interpret biological information. And the field of bioinformatics is, we can say, an interdisciplinary subject which has impacted medicine, which has impacted biology, physics, computational science, and even to some extent mathematics and statistics. And that's the reason that everyone's perspective for bioinformatics is different. And we cannot give a defined outline of bioinformatics per se. But today we will try to limit it for biomedical applications. We, being the human being, is one of the most complex system that exists on Earth, and the human being himself is composed of so many working organ systems and various organs, and none of these organs are spared with a non-disease condition. That means each and every organ in some phase of life or during any condition, they do have a disease condition. And that's how the whole field of medical sciences, or the field of biomedical sciences that exist, and they progress. And looking at the plethora of diseases, those are prevailing on human beings and other living organisms, the bioinformatician, they thought that how we can impact uh, that aspect. That means if there is a disease, people used to go to a doctor, they will visit a doctor and try to get some medicine and get rid of that. But then how and where a bioinformatician can really impact this process. So as we go down, so from the human to organ system to organs, tissues, and ultimately you end up with a cell which has got its own blueprint of the whole genome, which is there. And ultimately, that blueprint of genome, it consists of a set of genes which is which are linearly arranged, and ultimately each and every tissue in the body will have its own combination of genes which are expressed, and that's why a RBC is different than a brain cell, and it is different from a hepatic cell in epithelial cells. So that's how there is cell specific expression of various genes, and that really gives them the functional advantage of working as that specific cell. So in the central dogma of life, we all have read in our undergrad that the DNA is ultimately, through transcription, it forms mRNA and mRNA to protein, and ultimately this protein is the functional part of our genome. So the human quest for exploring himself, that means to sequence the whole human genome, it started way back in 1990, and it was the race was ended in 2003, where the first finished version of human genome was published simultaneously in Nature and Science. But if you look at the background of that, thousands of scientists, they were working day and night using the then available technology, which was called as first generation sequencing technology. And later on, this first generation sequencing technology saw the face of a second generation, where high throughput and parallel sequencing was feasible from different platforms. And now we are in the stage of third generation sequencing, where we look forward to sequence in real time the long read sequencing. So a whole genome of a small organism can be sequenced on one go. And during the whole journey of human genome project, say starting from 1990 to 2003, intermittently there were many small bacterial, viral, fungal genomes. Those were sequenced in and around 95 to 1998. And ultimately, we ended up with the whole human genome draft. And we thought that once we have the blueprint of the whole genome, possibly we will solve all issues related to human disease, human health, and everything. But once we completed that, we ended up with more complex situation, where instead of solutions, possibly we have generated few more issues and challenges, and that need attention with human intervention. But how to go ahead with that? And that's how the birth of bioinformatics came into existence, to support the further interpretation, further analysis, storage of these human genome data. Because these human genome is around say 3.2 billion base, and out of which hardly 20 to 25% are in the in the genes and related sequences, they cover around 25%, whereas 75% is in the intergenic regions. And we initially called it as junk genome, but nowadays we can find really some meaningful information from those intergenic regions, which is mainly composed of say around 55% repetitive and around 20% which is unique. And out of the whole 3.2 billion base pair, possibly 60 million base pair are really clinically relevant, and the whole race of diagnostic market in the clinical setup is somewhere around this 60 million cases. Immediately after the birth of the bioinformatics in to support the human genome. Few more omic technologies they came into existence. So uh, if you are dealing with, say, gene, and the complete set of those genes are the genome, then you are in the field of genomics. So omic become an suffix, a global suffix for the specific domain of biological sciences. So genomics, proteomics, transcriptomics, metabolomics, all these buzzwords, they came into existence in the post-human genome era. So in the genomics, people mainly deals with all the sequencing technologies. Mainly these are high throughput technologies where the whole genome of either a microbe to man, everything falls under this. Followed by microarray, which is dealing with mainly the transcripts. So sequencing, it mainly deals with either the whole genome or even the whole transcriptome. But microarray deals with the gene expression studies, where you see the differential expression of a diseased versus normal cell, or the effect of a treatment on a diseased cell at different time interval. So it shows a relative gene expression changes in different physiological or pathological conditions. And these microarray, which involves a high throughput, means around 30 to 50,000 gene expression data sets per uh, individual experiment set. And then we analyze these microarray data sets, and we end up with a set of meaningful genes which are really responsible for the disease condition. And ultimately, we validate them with the help of a more specific and sensitive technique like real time PCR that validates the microarray predictive models. Similarly, in proteomics, it's not that high throughput as compared to the genomics, but still we deal with hundreds of different proteins, or even say thousands of different proteins in the biological fluid in different physiological and pathological conditions. So there can be few destructive and few non-destructive technique which deals with proteomics. And even these larger data sets, they are also being analyzed with the help of several dedicated tools or several online free tools. And we look at the functional change in these biomarkers in the disease conditions. Similarly, metabolomics is ultimately the flux of the functional system that is going on in the living being. And this metabolomics is more dynamic as compared to the genomics and proteomics, because genomics is the ultimate blueprint and only the variation is in transcript level. But metabolomics is very much dynamic, and it changes from different time interval in different disease conditions, in different phenotypes. And that's how it requires more sensitive as well as more specific way of detecting them. But if we only focus on the bioinformatics in healthcare, we usually starts with the disease condition. And for each disease conditions, the scientists who are engaged in this biomedical research, they try to find out and end up with a good and sensitive biomarker or specific biomarker for this specific disease. And once we end up with this biomarker, usually we try to look for a suitable drug for controlling this disease or eliminating this disease. And usually we try to find out a biomarker which is mainly responsible for causation of the disease. And then only we will be able to target it with some suitable drug. And so if we have a bunch of biomarker for a specific disease, we will try to find out that which is the most suitable marker. And if we develop an antagonist against that, we can control the development or progression of the disease. and that is the suitable drug target. So drug target identification is the next choice or next option for a bioinformatician to begin with. So the journey of bioinformatician, it starts from the biomarker screening identification, and then narrow down to the drug target identification and validation. And finally, once you have that target with you and you want to develop some antagonist or a drug against that, we look for the source of ligand. So whether you are going to search ligand from natural sources, or whether you want to explore the possible ligands from the drug databases which are already being approved by FDA, or the synthetic molecules which are developed by the chemist, synthetic chemist, and those are archived. So millions of such drug molecules are available across the internet platform, which are either paid or open source databases. So using those databases, as a bioinformatician, we go for in silico, or what we call it as virtual screening, using docking and simulation approach. Again, to do that, there are several paid tools, commercial tools, and there are several academic versions of these, which are free to use by for academic and research purpose. And ultimately, whatever tools and technique that you use, you end up with a few sets or few tens of those biomolecules, which can act as a potential ligand molecule for that specific target. And then now we wish to screen them in the preclinical setup, where we start with a few different cell line model, if it is a anti-cancer agent, and followed by animal models, two or three animal models, either a murine or guinea pig or rabbit. So all these animal models are part of the preclinical screening of drug discovery. And ultimately, if you end up with a good set of ligand which have shown good activity in the cell lines, and they have shown good activity in animal models with least toxicity to the different vital organs, then those molecules, they enter into the clinical trials. And there are various phases of clinical trial, so starting from phase 1 to phase 4. And during this, depending on which type of drug it is, the phases of clinical trials are opted. And ultimately, once the ligand or the drug like molecule successfully passes all the phases of clinical trial, then the drug is released into market for dissemination or for patient use. But even after the release of drug to the market, there is a post-marketing survey that whether the drug is equally effective against or across the population, or is there any specific population which are more sensitive or which are showing adverse reaction to the released drug, so that this can be further modified with a warning label that this drug is not suitable for this population, or this clinical situation. So that is called as the adverse drug reaction monitoring in the post-marketing survey. And then the role of pharmacogenomics comes into picture, that why those set of patients, they do not respond to the specific drug. And we look for, is there any genetic change in that specific population or that specific individual which makes them susceptible uh, for this drug reaction. And if it is so, then we may come up with a guideline that before going for this drug, we need to check for the specific mutations in the specific gene, or whether this gene is somehow inactivated in certain group of patients. And those pharmacogenomics warnings or labels can again be there in the modified drug. Or if the drug is showing some major reactions, then it can be thought of for a drug repurposing. That means the drug which was produced for a specific disease condition can be utilized for some other purpose. So one such example was the thalidomide drug, which was developed for the morning sickness in pregnant women. But later on, it was found that it has some defect in the developmental condition in the fetus, and birth defect was there in majority of children. So the drug was withdrawn. But later on, this same drug thalidomide was found to be suitable for some cancers. So that's how the drug repurposing also is done. So for identifying the disease targets, there are several set of disease databases. Those are available starting from Malacard, that is for human disease database, Nord, that is for rare disease database, OMIM is for online Mendelian inheritance in man. So it is a catalog of human genes and genetic disorders. Similarly, CAG disease database is also available, and disease GenNet. So there are lots of disease databases are available. And finally, as we discussed in drug discovery, the first step is in silico. Say before 20, 30 years, where the existence of bioinformatics was not there, the drug screening was done from the preclinical model. That means if you have say 1,000 different compounds as a potential drug, and you have to screen 13 different cell lines and 3 different animal model, you have to go through all those screening process in the wet lab, and then you will end up with a set of a good ligand. And that manual labor, later on, it was reduced with the advent of in silico technologies or in silico platforms, where you can screen millions of different drug-like compounds for several targets, and that gives a clue for a lesser number of compound to screen in the preclinical stage of drug development. And finally, it enters into various phases of clinical trials before FDA approval. And the last phase, as we discussed, it is the pharmacogenomics or genetics, which helps them. So, since we are going to discuss only the bioinformatics part of that, I would restrict the talk to in silico process only. So, in silico process of lead identification, it is mainly focusing on the biomarker discovery. So, as I told, if you have only the disease condition known, and if you are going to identify a new biomarker for that set of disease or the disease condition, then our first approach will be to go for a high throughput technology involving next generation sequencing or microarray, where the differential gene expression profile will give you that specifically which set of genes are overexpressed in disease conditions as compared to the normal. And from those set of target biomarker, we can further go for a pathway analysis where we see that each of these biomarker, how they are involved in the metabolic pathways, which is responsible for survival or progression of these cells, and that is responsible for causation of the disease. And once we end up with a hub node that is interacting with more than one partner in the metabolic pathway, possibly that can be a suitable drug target. So target selection and validation is also possible through bioinformatic tools. And once we end up with the right target, possibly we call it that as the suitable target, and we prepare the target molecules. Many a times it's not feasible that you have the three-dimensional structure of all those proteins. Unless it is well characterized, possibly you only have the sequence of the protein, but not the three-dimensional structure. So there are databases where you can identify and you can download the three-dimensional structure of the target protein. And with the help of the drug library or the natural compound library or small molecule library, you can go for identification of a lead ligand against the target that you have selected. And that part of bioinformatics is also called as structural bioinformatics, which is based on the proteomics aspect. Right. So if you look at that, there are two possibilities. For virtual screening, it's feasible when the target is known. That means if with the help of say microarray or with the help of other published literature, you could end up with a suitable target. So when the target is known, we will try to find out whether the structure of that target is available or not. If it is available, you can directly download it from the protein data bank. But if it is not available, there are tools in bioinformatics which can predict the structure of the target. And then you start your virtual screening using either natural, synthetic, or customized ligands. So it is ligand based drug discovery. Or there are other facets like structure based drug discovery or fragment based drug discovery, which gives a potent ligand for that specific target. Many a times the target is unknown, and then you really take the help of the various high throughput techniques, and then you go for hub or the target identification. And as I told, if the structure is not known, you can predict it, and ultimately you can again go for the virtual screening. Immediately after the screening, so this molecular docking study is like a lock and key where the target protein is acting as the lock and all possible keys are possibly your ligand. So the key that best fit into the key can be a possible key for that specific lock. Similarly, the process of virtual screening will shortlist few of the ligand from the archive of say millions or thousands of drug-like molecules. And then the docking results, which shows the interacting ligand with the binding site in the target protein. So these are all in a rigid conformation. So the best fitting ligand can be further processed for molecular dynamic simulations, where you provide flexibility to both target protein and the ligand for a vibrational motion. And then the most suitable or most stable ligand, which still remains in the target fitting to the binding site, is possibly the best ligand, which can be later on converted into a drug. Then once you select a set of ligand molecules, the ADMET prediction. So ADMET is absorption, distribution, metabolism, excretion, and toxicity that is required to label the ligand as a drug-like molecule. So usually these tests are performed in the in vivo conditions in animal model, but there are now tools available in bioinformatics to do ADMET prediction of all possible ligand, and then the ligand with least toxicity and better bioavailability can be taken further as for preclinical and clinical trials. So the drug likeness, blood-brain barrier analysis, and even off-target binding. So the molecular docking studies shows that the ligand fits better to this specific target. But what about this ligand goes and bind to other thousands of drug binding proteins in the body, and how and where it can bind and fit. So that is also required before you label them as a good drug. So then the lead confirmation is usually done through in vitro screenings. So it is a cell-based assay where the cells are plated on microtiter plates, and you look for their cytotoxicity. If it is for cancer, we will look for their anti-cancer activity. So there are different development in this phase, like screening of organoids or spheroid cultures. So that gives or that mimics the three-dimensional nature of the tumor in the body. And ultimately, the one which is showing best cytotoxicity with less concentration can be the suitable drug-like molecule. And then these lead validation is done in the in vivo system in either C. elegans or some rodent model or non-human primates. So there are different ways to validate this. Where if it is say for cancer, you can grow the cancer cells through xenotransplant on uh, whether rodent or guinea pig or even in primates, and you inject the compound of interest and you see how the tumor size is decreasing or not. So all those falls under validation, and finally the compound which is effective against the disease and with least amount of acute or chronic toxicity, it enters into the clinical phases where starting from phase 1, phase 2, phase 3, and then the FDA review and it is approved. And in the phase 4, possibly we look for post approval surveillance in different population. And that pharmacovigilance, again, it is associated with genetic variation in either the drug metabolizing enzyme or in the target molecule. Say if there is a mutation in the target, and due to that there is a structural variation in the target molecule, and that doesn't allow the drug to bind to that target effectively, and that's how the cell survive from the effect of the drug. So all those predictive models are again done by the bioinformaticians. So once you have the information of the genetic mutation, insertion or deletion, how those affect the structure of the target which we are aiming at, or whether the drug is metabolized before it reaches to the target at the phase 1 detoxification in the liver, or whether there is some obstruction in its absorption. So all those are finally being taken care during the pharmacovigilance survey. As I told, there are two major aspects. One is structural biology, and second one is based on the genomics or high throughput technology like next generation sequencing and microarray, which helps in identification of the target molecule. So there are tools like bioconductor, GenSpring, and even GEO has its own tool where we have the microarray archive sources where the microarray datasets are available. Like GEO is one of the major source who archives the microarray databases. Similarly, SRA databases are there for sequence retrieval, and these are deposited with microarray, different microarray platforms across the globe, and they deposit their raw data files in this, which has we may have access to that for further analysis. And using these, there are few paid tools like GenSpring is a paid tool, and there are free tools like GEO2R, Bioconductor, which are based on R. And we can use this technique and do the differential gene expression studies. So this is just a pictorial diagram of how a microarray chip is embedded with thousands of the complementary sequences of the small fragment of around 30 to 60 bases, and these are imprinted in multiple copies. And when you put your labeled cDNA in that, and binding of this will give a fluorescence intensity, which is measured through the laser-based scanning system. And from this, we can come to know that which are the set of genes which are overexpressed and which are the set of genes which are downregulated in diseased and normal conditions. And ultimately, using those tools, we do the normalization, cluster analysis, classification, and finally the pathway analysis of the highly upregulated and downregulated gene, which uh, we can use them as a possible drug target. Similarly, for an NGS workflow, it begins with, say, isolation of nucleic acid. If you are going for whole genome, possibly you are beginning with the DNA of the specific diseased tissue or cell. And once the nucleic acid is isolated in the purest form, then you go for fragmentation, because the second generation sequencing platforms, it deals with smaller fragment of nucleic acid from say 50 to 500 base pair. And since it is a high throughput technology, multiple number of samples can be processed parallelly. And that's the reason we need to label them with some barcode so that the sample mixing is prevented. And to do a bigger, deeper depth of sequencing, we need to amplify the genome or the nucleic acid content, so that the same region of interest can be sequenced multiple times. So 50x, 100x, 500x, so that x is the time of number of times the same region of nucleic acid is sequenced. And once the sequencing is done, you will get the raw data file depending on which platform you are using. And then we have to perform on the data analysis, and that shows what is the situation, where are the indels, insertion, deletions, duplications, or if it is, say, transcriptome, then it is copy number variation and all those. And usually, with each fragment of nucleic acid, there is a primer and followed by an adapter, which is very much specific for that specific sample. So now there are adapters available for around say 96 number of samples, which can be multiplexed, so that they have got a unique barcode on their end. And this is useful when you want to do the data analysis. And before that, when we are aligning all the sequences, you do demultiplexing, so that each sample data is segregated into a separate file, and then you align them and look for what are the variations in the whole genome. So this is also a brief workflow for NGS data. So you generate raw data from the sequencing platform, whether it is iTorrent, whether it is Illumina, and then you do the initial quality control or the QC informations which are there along with the sequence data. Then you try to align and you identify the possible indels, and that can also be visualized in the form of the charts and plots. So ultimately, whatever you perform, and depending on the need, whether you are a biomedical researcher and who is looking for some new biomarker, or whether you are a bioinformatician, structural bioinformaticians, who starts from the target and end up with some probable drug-like molecules, which can be taken further by a pharmacist or a pharmacologist to convert it into a potent drug-like molecule along with the help of medicinal chemistry. So, as a bioinformatician, we have possible areas of research collaboration with a medical setup or biomedical fraternity. So, whether you start working on communicable diseases or like pathogenic organisms, we are now possibly facing the challenge of COVID exposure or COVID pandemic. So globally, all workforce in the research field or in diagnostic or in therapeutics, everyone is focusing on mitigating the COVID pandemic. So these are the possible areas where, as a bioinformatician, we can contribute. And I think there was an upsurge in publication in this last 2 years, specifically on COVID. So thousands of literatures we can see. Everyone is trying to do something on the COVID targets. So there are several non-communicable diseases also, which are chronic and long-going. So diabetes, hypertension, arthritis, obesity, all these, even cancer is part of that. There can be neurological disorders, starting from, say, uh, degenerative changes like Alzheimer or any other neurological conditions. There can be a work on birth defects. And if you are a toxicologist or a forensic scientist, possibly you can look for the cause of death analysis based on this, and many more areas to collaborate in the field of biomedical research. But whenever we are trying to collaborate in the biomedical research, there are role of segregation of role for both of us. So as a clinician, they may have role as generating the hypothesis. So what is the research questions for them based on the observation of the patients and the clinical instinct that is being developed with their experience. Approval of ethics committee is also a part of biomedical research. Whenever you want to begin with and you want to sample the biofluid from patients, we need ethics approval. Patient selection, clinician can guide. Clinical data management and correlation with research data is possibly their role. And writing a publication is partly by both the collaborator as a clinician and as a scientist. But as a scientist, we have role in designing a study to address the question which is being raised by the clinical colleague. An approval from scientific research committee is our role as a scientist. And then we perform the sample analysis. So whatever sample that is collected is analyzed in the lab, and analysis and interpretation of data is also our domain or our role. And again, we share the contribution in writing the publication. So that's how there is a balance of clinicians and scientific fraternity for collaborative biomedical research. I think this is for today. So now I would expect a few questions from the students.
Harshida,
Yes,
please read out the questions from the.
yes, yes. Whether targeting gene by microRNA constitutes same effect as that targeting transcription factor by microRNA.
So when you are trying to target a transcript, there are two possibilities. So whether you are trying to target the mRNA which is there, and if it is say mRNA, then possibly targeting them with a antisense nucleic acid or a microRNA is a suitable option. But if you are trying to control the expression of that specific gene, then possibly transcription factor binding site, targeting the transcription factor binding site can be a better option.
Harshida, you're please unmute yourself.
Unmute yourself.
Okay. So another question is, what will be an appropriate way of studying proteomics of drug discovery after the identification of SNPs and indels?
So, if you have the SNPs and indel in hand, then we can definitely go for in silico mutagenesis, where you have your wild type protein and you try to make the mutants out of that. And with the mutant also, you can try to screen the ligands. And if you can find a ligand which is working well against the wild type as well as mutant, then that is possibly a suitable target, a suitable option. Otherwise, we can have a different set of ligand for mutants. And that's how the several versions of therapies are coming up. Say, if you take the example of the BCR ABL target, where people are targeting the ABL kinase domain. Imatinib was the wonder drug once upon a time, and that was the gift of bioinformatics. It started with the bioinformatic approach. But now there are later version of imatinib, because the kinase domain of ABL is facing many mutants. So there are a lot of mutants in that, and that is resistant to imatinib. So that's how from imatinib to nilotinib to dasatinib, and dasatinib to ponatinib. So different versions of the drugs are coming up. So there is always a scope for screening a new set of ligand against the mutants if the information is available. Otherwise, we have to look for it in the pharmacogenomics.
Okay, so we have another one question. Is there any computational method available for correlating novel miRNAs or predicted miRNAs and disease? Kindly discuss the strategy.
So if I get time, I can show you one of uh, the study of my student. So there was one study of Dr. Kinjal Bhadreja. She worked on Moringa microRNA. So when we did the sequencing of microRNA from Moringa oleifera, so that is drumstick, and we could find several differentially expressed microRNA, and which were unique for this species. So there are tools which you can identify the microRNA which are specifically found in those species. And then there are tools which helps in predicting the sequence as a possible microRNA, whether the hairpin structure is feasible or not. All those criteria, there are different set of tools for that. And then you end up with the real microRNA which are possible both structurally, and then you can move further for predicting their targets. So when you blast them against the mRNA of human targets, possibly you will find the suitable targets which these mRNA, these microRNA can interact with. And later on, using all those protein-protein interaction and hub node identification approaches, we can definitely end up with a potent suitable target.
Thank you, sir. So, thank you so much, sir, for such a great and valuable session. I'm sure this session will boost participants' mindset and working practices. We had announced a poster competition on two themes from PG and research scholars, and I'm happy to share that the results are ready. I request Archana ma'am to announce the results of the online poster competition.
Thank you, Harshida. Like you mentioned, an online poster competition was announced on two themes. Theme A was computational genomics, and theme B was computer aided drug discovery. We are happy that PG students participated wholeheartedly for this, and I'm happy to announce the results. So, Young Scientist Award for Theme A goes to three participants: Chaitali Pandya, Mohit Giri Goswami, and Jensi Thumar. And Young Scientist Award for Theme B goes to Shweta Tiwari. So, congratulations to all the winners.
Okay,
Oh, thank you.
congratulations to all the winners. Now I request Maitrai sir to present the formal vote of thanks.
Thank you, Asudha. Respected Professor Dr. Himanshu Pandya, Dr. Jagdish Bhavsar, Dr. P.M. Patel sir, Dr. Rakesh Rawal sir, my colleagues and dear participants. At the outset, I thank Professor Dr. Himanshu Pandya, Dr. Jagdish Bhavsar, Dr. P.M. Patel for always encouraging us and blessing us. I also thank our distinguished speaker, Professor Rakesh Rawal sir, for sparing his valuable time for us and giving a very nice overview of signification of bioinformatics for healthcare. I thank the judges for sparing time and helping us in evaluating the posters. I thank all the participants for their enthusiastic participation in today's webinar. I thank Dr. B.S. Agrawal sir, Director Rolwala Computer Center, Gujarat University, to facilitate live streaming of the event on university Facebook page and YouTube. I thank Mr. Sagar Panchal from GeoSec for technical help. I also thank Ms. Arsida Gadwe for anchoring this webinar so gracefully.
Thank you,
And
sir.
last but not the least, I thank my colleagues in the department for their timely help. And I congratulate all the participants and the winners of the poster presentation. Thank you. Thank you very much.
Mm, thank you, sir. With this, we come to the end of this session. We have had a very informative and exciting learning experience, and I'm sure all of us will now be able to relate to the role of bioinformatics for healthcare. I once again thank Raval sir for sharing useful information with us. Thank you. The feedback link is on the screen. Please fill it up and receive your e-certificates. On behalf of Department of Botany, Bioinformatics and Climate Change Impacts Management, Gujarat University, I once again thank you all for your participation. Thank you.
Thank you.
Thank you.